Hi everyone, I'm Elna Gottlieb from the DNA Learning Center, and welcome again to another session of DNALC Live. So today we're going to be studying human variation. And so we're going to be looking for the differences. Variation, that means differences. We're going to be looking for the differences between humans. Now, to understand those differences, we really need to understand how much we have in common with each other. So you need to know your variation, you need, well, what's the commonality? And so for that, you have to kind of look to the DNA. Now, we're basically the product of our DNA. And so the DNA, this is like the grand cookbook of us all. So this is how we get our traits. And in that DNA, we have genes, and those genes will produce those traits individually. Different genes will control and produce traits. And so therefore, if we compare all of our DNA, so if we take that DNA and compare it to any human that you're near, any human you're sitting next to, you're going to find out that you have 99.9% in common with that person. Now, if you happen to be sitting next to an identical twin, like your identical twin, well, then you have a lot more. You have nearly 100% the same. If you're sitting next to a sibling or a parent, then you're definitely going to have more than 99.9, but probably not quite as close as an identical twin. So we share a ton of DNA. So there's 99.9% of our DNA that makes us exactly the same. Now, on the other hand, that leaves a tiny amount of 0.1%. All of our differences lie inside that 0.1%. So that's a tenth of 1%. It didn't even make it to a whole percent. So that is a tiny amount. And it makes it really interesting to study these differences. And so we're going to take an opportunity and we're going to study some of these differences today by looking at traits. So we're going to take a look at traits and we're going to see our differences. We're going to see in places where we share things with each other. We're going to be testing out a few traits. We'll see how genetic they are and how, some things maybe aren't as genetic as you think. And we'll also, or at least I, will be tasting something too. So I'm going to taste something in front of you. You'll have the opportunity, or at least I'll tell you where to get this thing. I'll tell you more about it later. And so now, first I'm going to take you to my screen. I'm going to share my screen right now. So I'm going to pop off of this camera. So I'll see you on the flip side. Okay, so here we are. So to study human variation, genetics, just to let you know, genetics, let me just change my color here. That's the study of DNA and genes. There we go. But there are two really important words that we're going to come across a lot. And so these two words are genotype and phenotype, or in this case, phenotype versus genotype. And so you do need to know the difference between those because they come up a lot in what we do, especially in our activity today. So first, I'll make it kind of easy, and I'll start with genotype because we've been talking about genes a whole lot. And so, Jen, it has to do with genes. So when we're talking about genotype, we're talking about the actual DNA. So the actual DNA, the actual genes responsible for making or building traits. And so now those genes, that DNA, you inherit from your mom and dad. So you get half from mom, half from dad. So therefore you're getting genes from both parents. So you can receive genes that will give you a certain hair color. Let's say your mom gives you genes that, involve the color blonde. Dad can give you genes that involve the color brown. And so you will receive hair color genes, but just two different variation. We actually call variations or types of genes, we call them alleles. So you'll see that a lot. That just means you have a gene for hair color. Actually, you have a lot of genes for hair color. Hair color is a little complicated, so we won't cover that today. But you have genes that create a certain hair color, and then you get another set of genes that create a different hair color. Those are alleles. They're creating the same trait, but they have variations that change the trait a little bit from person to person. And that's where genotype is important. So it can kind of blur the lines a little bit when you're looking at phenotypes. Now, phenotypes, on the other hand, has to do with the product of your DNA. So we're talking about product. And so we're talking about what the DNA does. And so that DNA, it causes your traits to be expressed. So when we're looking at a phenotype, we're looking at the expression of a trait. And so therefore, you may have certain genes from mom and dad that give you a certain hair color, but that hair color is the actual phenotype. So that brown hair or blonde hair is your phenotype. That's going to be important because you're going to see sometimes phenotypes can help you figure out a question in genetics. Sometimes they can blur the lines in genetics. So we'll study that when we come back to it. And so from here, what I'm going to do is I'm going to come back to the camera and we're going to go over some traits. So I'm going to give you some examples of traits, and I'm going to go through these traits, explain what they are. And by the way, you do have some pages in the resources section. So if you go to DNALC.org and look under studying variation in humans, or human variation rather, then you'll see that there's a little resources tab where you can download and print that stuff, and or look at them on the screen if you wanted to. Otherwise, you could just follow along with me, that's fine. All right, so I'm going to come off my screen share right now, and you're going to see me in just a moment.
Okay, so let's look at some phenotypes. So I have this little human traits checklist. So these are the phenotypes we're going to look at today. And so they're simple. There are some questions whether or not they are fully genetic. There's a lot of questions actually, but they're still traits that you can see. And so these are traits that are observable with your eyes, except for one, which I'll go into that in a minute, or quite a few minutes actually. And so we're going to go into them. So now I'll go through what I have, and then I will show you some examples of what I don't have, and you'll be able to follow suit if you have this list and see what you have. So the first trait is a cleft chin. So if I put this up here, right here, cleft chin or no cleft chin. And so, well, your chin, yeah, it's right here. And so a cleft is when you have a dent in your chin. Now you can have different forms of a dent. You could have something that looks very much like a dimple, like it's almost like a hole in your chin. And there's a famous actor, he actually just passed away recently, Kirk Douglas, who had a deep hole. It was sort of like his signature mark. But you could also have a dent. I have a dent. And so it has this little dent, so it's not rounded right here. It's got this little dent. It can be subtle. And so if you put your fingers here, relax your face a little bit, and feel like the two sides are a little higher, then you might have a dent. If it's rounded, then you do not have a cleft chin. So I do have a cleft chin. I don't have a good example of a non-cleft chin. I just don't have a good picture of it here. But for now, I am going to highlight cleft chin. So I'm going to highlight cleft chin on my human traits list right here. All right, the next one is widow's peak. Now I have a couple of good examples of what a widow's peak looks like. So first of all, my hair. So if I work my hair back, you're gonna see that I have a little dip. So I have this little dip right there. That is a widow's peak. It peaks right usually in the center of the forehead. Sometimes it's not perfect. Mine's a little kind of just pointing a little crooked, but it's still sort of in the middle. And this young lady here has a really pronounced one. So you can see that point comes all the way down. And so if you pull back and look in the mirror at your hairline and you see that it just goes straight across and there's no defined point like mine, then you do not have a widow's peak. So I do. So I am going to highlight my widow's peak trait right there. And so there we go. And so now the next one, free earlobe. Free earlobe and detached earlobe. And so, yeah, this is a fairly famous trait, but it doesn't mean that your earlobes are detachable. It means that they are connected. But what it is that your lobe hangs down below the connection point. So mine hangs down a little bit below, and that's on both of them. And if you can tell on your ears that it both, this one I think hangs down a little bit more than this one, but this one still hangs down. So I have free earlobes. Now I'll show you a good picture that shows you the difference. So right here, yeah, this guy's a little bit funny. But if you look at this middle picture right here, then you'll see this ear hangs very low from the connection point. So let's say it connects right here, it hangs very low. So that is a free earlobe right there. Now here, this ear kind of goes right to the person's neck. So that's attached. Even though it's not that the earlobe's gone, there's an earlobe there, but it just attaches straight across. So if your earlobe goes straight across and doesn't hang lower, like this is extreme, but if it doesn't hang lower than the attach point, then you have attached earlobes. Now they used to say that this was a specific genetic event. And, but, you know, when you start looking at lots and lots of people, you start realizing, why does one person have one attached and one not attached? And so there's a lot of fuzziness into how these traits come about. So, and we never could really nail down exactly a genetic gene that controls whether or not your earlobes are attached or not. But they're still a good trait to look at. So it's still fun to assess them and compare them between people. All right, so now I have free earlobes. I have both free earlobes, so that makes it easy. If you have one attached, one free, see, circle both. That's fine with me. All right. Now this one, remember I said I was going to taste something. And so this says to taste PTC paper. So we're going to skip that for now. We'll come back to it later. We've got a whole episode sort of on that. Well, not a whole episode, but we've got a whole separate taping for that. And so you'll see. And so we'll get back to that, and I'll tell you what it is. So don't worry, you'll find out a lot more about it later on in the session. And so I'm going to skip to the next one. Straight thumb versus hitchhiker's thumb. Now, right here, this is a pretty good picture of a hitchhiker's thumb. And so what it is, it has a bend back, so it bends back. I have a good example of more of a straight thumb, so it really doesn't bend back. It curves slightly. Maybe this one, no, they both kind of curve the same, but it's still fairly straight. It doesn't really have a good bend to it. And so I kind of consider that more on the straight side of thumbs, whereas this person right here has that curve back where it literally curves back. Some people can actually do like a right angle almost, real crazy curve there. And so they have hitchhiker's thumb. I don't consider myself having a hitchhiker's thumb. And so I am going to highlight straight thumb for me. So now this next one, bent little finger. It's a little trickier. It has to do, you have to look at your fingers, and you have to look for the little finger, which is your pinkies. And what it is, they bend towards the other fingers. Not everybody's does, but that is a trait that they've seen. And so if you can see it in this picture, it's not too bad. The pinky, you can see it's kind of bending towards the other finger. So that's when they say a bent little finger, that's what they're talking about, where the tip sort of like curves in towards your other fingers. Now there's a couple ways to tell this. If I take my two pinkies and put them like this, you can actually see that they curve away from each other a little tiny bit. So they curve away a little bit. And so especially this one has a little bit more of a curve than this one. And so my pinkies are pretty straight, but because of that little curve, I'm gonna say that they're bent. I'm just gonna, even if, you know, somebody comes and argues with me, I will say that I have bent little fingers, even if it's slight. And so there we go. Now, the next one is always tricky. It's mid-digit hair. Now, to understand mid-digit hair, you need to understand where your mid-digits are. A lot of people get this confused because they hear hair and they go to the one part of their digits that has hair, which is this part. You're always going to find hair. I don't know if you could see it on mine, but yeah, you can see the little tiny like holes, sort of like those are budding hairs that are coming out of that part right there. So all the way across, my ring's in the way over here, but that's a little bit of hair, very little hair, but there's, you know, some people have more hair, as you'll see in a second. But that's not your mid digits. So the part, like if you make a fist, not that I want you to make a fist and get into fights a lot, but if you do this, that's not the part we're looking at. We're not looking at the hair that's closest to your biggest knuckles. We're looking at the mid digit. So you'll see, oh wow, there are actually 3 little parts of digits right here. And so the mid digit is the center section all the way. So you'll see that your thumb does not have a mid digit. There's no mid digit there. It's got 2 digits, that's it. And so this is the mid digit. Now, if you're looking for mid digit hair, it is most commonly found on the ring finger. So on your ring fingers, mid digits is where you can look, and that looks bad, but it's not. So you have, if you look here, and so if you don't have hair on those mid digits, then it's unlikely that you'll have hair on the other mid digits. But that's the first place to look, but it's not, you know, I'm not going to say it never happens. So there's always exceptions. So you're going to look, but that's the first place to look, because if you do have it, it's going to be most visible on here, most likely. And then you can look on the others. The middle finger has one too. And so I'll show you over here. This is a pretty good picture. This guy's hand, there's a lot of hair on there. So there's lots of hair on that first digit right there. But when you go to the second digit, this one right here, clearly you can see mid-digit hair. But if you go a little bit closer, it's a little hard. The light's a little yellow, but you see these little dots right here. So he does have some hair on the mid-digit, on the middle finger right there. He's got a cut right here. I can't tell if there's hair. I don't think there's hair on his first finger or his pinky. So basically these 2 fingers have a little bit of hair. This one has more than the other one. And so if you have a little bit, you might just be able to see these little, sort of like little dots. These little, they might be almost invisible. So it's a little hard to tell if you have that hair. But you do the best that you can. I do not. And so I don't have mid-digit hair. So, oh wow, look at that. It's the first one on the opposite side. So I'm going to highlight no mid-digit hair. Okay, so the last one on our page we're going to look at is dimples. Now, there is one more. I'll come back to that one. But let's take a look at dimples. So now dimples, this is fun because this is where you have to smile. Now, I don't have dimples. My face is dimple-free. There's no dimples because I do have creases. That's different. So creases, everyone has kind of these creases. Sometimes you have more than one crease. Those aren't really dimples. The dimples come a little bit behind. Sometimes people have dimples in unusual places, like a little higher up. Sometimes they have them a little bit lower down, a little more towards the chin. And so if you have any dimples in odd places, feel free to record that on here. That's fine. That's neat. And but if you have them in the usual places, then you can put dimples. Now I'll show you an example. Since I'm not a good one, I have none. And so this lady here, she has very well-defined dimples. So you can see she's got these little dimples right here, and they're in the typical place. What a dimple is when the skin actually gets kind of stuck to your bone. And so as you grow, your muscles develop. Then when you smile, that skin gets pulled in a little bit. So that's what a dimple is. It's just like a natural connection point that isn't always there for everybody. And so she's got some nice defined dimples right there, and you may too. Are they always genetically inherited? Well, they can be. My mom has one on one side, so I definitely didn't get that. I have no dimples, and so there we go. And so I have no dimples, so therefore I'm going to highlight no dimples. There we go. So no dimples for me. Not even one. Okay. And so now we're almost to our next stage, but there's one that I haven't done yet. And that's this mysterious taste PTC paper. Now, you're probably wondering, a lot of you are probably wondering, what is PTC? That mysterious 3-letter word right there. We're going to go into that later, so don't worry. I'm going to explain it a little bit better. But basically, what we're going to do, and I have two, I'm going to kind of pull a fast one very soon. I've got 2 testers. I actually have the PTC paper, which has a certain taste to it. So PTC has a distinctive taste that only some people can taste. And so some people don't taste it at all. Some people taste it. Some people taste it very strongly. And so I'm not only going to test it on myself, I'm going to pull my family in, and we're going to test it on my family. But I'm going to pull a little faster on you. I'm going to let you know. I'm going to let you in on it. And you'll see me show you this later. So remember this in our clip. I'm going to give them some control paper first, which means that there's no PTC on it at all. And we're going to see. We're going to see what their reactions are compared. And so it might be pretty tame. I don't know. But I'm going to trick them just a little bit. Just in the sake of science. We're going to trick them just a little bit. And so that's coming up next. So I'll have to rearrange my camera angle for that. And so we'll get to that momentarily. Hello, I'm back. I had to change my camera angle, and I'm also going to invite my family in. So my husband and my 2 kids are going to come on in because they're going to help me out by testing PTC paper. And so they have their papers already. So I've got it right here. And so they're going to put it. So here's what you're going to do. You're going to take the piece of paper and you're going to put it in your mouth. So if you have this at home, this is what you're going to do. And you're going to just use your tongue, kind of push on the roof of your mouth and just taste it. So like this. Don't eat it.
Come on.
Just taste it. All right. Now come on back in. They lost it for a second, don't worry. All right, go ahead. Go ahead and taste it and tell me your impressions.
Tastes like paper.
Tastes like paper.
Tastes like paper.
Okay, so it tastes like paper for me too. Well, actually, so that was the control paper. So it was paper. So I wanted you guys to see what the paper tastes like first. All right, so now we get the real stuff.
What?
And so here's the real stuff. It was trick. Yep, it was trick. So here's the real stuff. Now you guys are going to, as soon as I get this open, there we go. Okay, here's one for you. Here's one for you. Here's one for you. And I'm going to do it too.
Bitter.
Paper.
Yeah.
All right. So for me, yeah, it tasted bitter. Like, it tasted like a Tylenol stuck on the back of my throat. Okay. So now how about for you? What did it taste like?
Bitter. I can taste it. Whatever.
Bitter?
Yes.
Strongly bitter?
Yes.
Okay. How about you? Horrible? You want to go wash your tongue off with soap?
Can I?
Yes, you may. Go ahead. She's going to wash her tongue off with soap. How about you?
Just paper.
Just paper. So you didn't taste anything bitter?
No.
Okay. So we have 3 tasters and 1 non-taster. So we're going to analyze these results next. I'm going to show you how to analyze these results. And we'll assess this and see what the story is.
All right, so we're back to my screen now. Here is a better view of all my personal choices, or rather all my personal traits. And just to remind you over here, these are the phenotypes. So all of these are phenotypes that we're describing. We really don't know much about what's going on with the genotypes or the genes involved. We just know that these are traits. And so here's my list. So wow, I have a lot on this side. So there's a lot going on there, and only 2 over here. And here's all our people again, just to show you our group of people, just for fun. All right, so now, this is fun to do, and but how fun is it to just get a list for yourself? So to just do it yourself, and so you'd want to spread this out. You'd want to find out, you'd want to compare yourself to other people. So now imagine taking this list and handing it out to, let's say, your whole grade or even your whole school. Now that might be fun because you get back the results. You're going to get this list right here. But that's a lot of work. If you look, we've got 1, 2, 3, 4, 5, 6, 7, 8. We've got 8 traits that we're going to have to decipher. So we're going to have to look at everyone's answers and figure out where they fit among those 8 traits and those possibilities that you have right there. And so that makes it a lot tougher, a lot harder to wade through all that information, all that data. So there are some ways to make it simpler. There are some ways to make it fun. So here's a really creative way that we like to make it fun, or rather, here's an interesting way right here. Now this looks really complicated. You have this big circle. You see like the names of traits in there, and you have these numbers on the outside. Kind of looks like a really complicated puzzle. And but for me, it's a really fun way to kind of keep track of your data and the way it works. So now this is in the resources. So if you go to the original www.dnalc.org page and you go to the studying human variation click, if you click on that, then you're going to see in resources you have these pages, and so you can print them out. And to do this, so the way you do it, so basically you have to start in the middle. And so this circle that's in the middle right here, you have to start in the middle and you have to ask yourself that question. So the question in the center of our circle is whether or not you have mid-digit hair. You'll see the top of the circle, that hemisphere, that half a circle right there says no, the bottom says yes, kind of upside down. Now me, I've already gone through my traits, so I know that I have no mid-digit hair. So basically, I want to color that section in, but I don't want to color the yes section. So I'm just going to color in my half circle right there. There we go. So there's my half circle. Now what that does is that completely eliminates the bottom half of the circle. None of those traits apply to me because they all are connected to that mid-digital hair trait. So that means that all the numbers in between 8 and 25, those are not my numbers. So my numbers are from 9 to 24. My numbers are somewhere in there. All right, so now I have to look up the circle. So the very next one, I'll change my color. Change it to blue. And so the next part of the circle right here, so we're not looking at the whole circle all the way around, bottom half is gone, is whether or not I have a widow's peak. Now we went through me, I do have a widow's peak. So I don't want to color this, I'm going to color yes. And notice I'm continuing from that mid-digit hair. So I'm continuing out from the mid-digit hair. I'm not going to go and color in this one because that's in the part of the circle that doesn't apply to me. All right, so now we look at the next one. I'll change colors again. Go to purple. And so now we have this free earlobe section. But you'll notice I stop right there because if I look at these two characteristics over here, that doesn't apply because that's connected to having no widow's peak. So now my choices are here. You can see I'm starting to narrow down my numbers too. So it only goes from 9 to 16 now. My numbers are somewhere in there. Now, free earlobes, I happen to have both. If you're a person who looked in the mirror and said, oh, wow, I have one that's connected and one that's not, for a test like this, you might want to just pick the one you feel represents you. And so therefore, like if you feel like, wow, I'm really proud of my attached earlobe, then go with it. Go for it. It's all right. Not the end of the world. This is you. And so what represents you is fine. All right. So now that means that I've basically eliminated 9 through 12. And so now I know that my numbers are between 13 and 16 there. So now I'm going to change my colors again because now I'm going to the cleft chin. Now, I pointed out my cleft chin. So therefore, this is me. This is me. All right, right there. And so now I've narrowed it down to 2 numbers. Now, some of you, if you have done this test and you don't have PTC paper, well, then, you might be stuck in this situation where you don't know which number. That's okay, because you can still gather information. In fact, you can gather the information from everyone. So that would be your 2 numbers, 13 and 14, like them. Well, that's mine. If I didn't taste PTC or I didn't have any PTC paper, I would be a 13 and a 14. Now, I happen to have PTC paper, and so I did taste it. And so therefore, that makes me lucky number 13. Okay, so now you might be thinking, oh, wow, 13. So what does that mean? Am I special? Am I smart? No, not really. So these numbers just represent a pathway of traits. And so therefore, the 13s have a collection of these traits. That's what that is. So all the people who are 13 like me are going to share those traits in common. And so what's fun, having it in a small group is not as nice as having data from a whole big group. Like, for example, a whole class or a whole grade or even a whole school. And so that's kind of fun to see how many of these individual numbers there are. And in a small classroom, you might find that it's heavy in one direction. Like, for example, there could be a lot of 22s or 23s. For some reason, I find a lot of 23s in my classes. And but when you do the whole school, you'll find out, you might see that as the population gets bigger, these numbers kind of spread out a little bit more. So it's a really interesting way to look at data and study a fun group of traits. And so now that's my wheel. Okay, so now we're going to talk a lot more about PTC. I promised I would. And so we're going to talk about what that is and what it stands for. But first, I do want to record our results from our taste test. So before I forget, I want to write down. So now, first of all, me, I'm Elna, and I am a taster. I could taste it. So I'm just going to write that on the taster. My husband, his name is Mike. He is also a taster. He said he felt he was a strong taster. So I'm just going to write strong taster. My daughter, Emily. She also said she was a strong taster and wanted to wash her tongue with soap, which I hope she really didn't actually do that. But yeah, I don't really think she did. Anyway, and the last person, my son, Alex, he said he was just tasting paper, and that's it. So I'm going to call him a non-taster. So I just want to record those events so that we don't forget that. So we're going to kind of reflect on that. Now let me tell you a little bit about it. And so PTC actually stands for phenylthiocarbamide, and it is toxic in high doses. I think I've heard that the synthesized version in the laboratory can be very, very dangerous in high doses. That's what I've heard. And they used to do a lot of tests with it a long time ago. I think since then they found out. So what they give in these test kits are not as intense, so that it's not as toxic, although I wouldn't eat them, but it's fine for tasting. Originally, they do come from different types of plants, and yeah, it's toxic. It's got that kind of bitter taste to ward off animals, so it's trying to kind of warn the animal not to eat it. Now, another name for it is phenylthiourea, and so that's called PTU for short, and it's essentially the same compound, and so again, also found in different plants. Little fun fact, and so PTC, scientists found out that it inhibits melanogenesis. Now this may, be mysterious, melanogenesis. Melano melano in Greek means black. And so in this case, we usually use it for pigment. And so color, like dark color or dark pigment or an intense pigment. And so that's where that comes from. Genesis is to form or something that happens from the beginning. And so genes are considered like the beginning of the whole process of going from DNA to life. And so that's where that comes from, or genesis, the beginning of something. And so melanogenesis, so essentially inhibiting, which means to interrupt or stop. You're stopping the ability of the fish to make color. And so these fish are transparent. And so we call that melanogenesis. I'll see if I can include a picture. I forgot to put one here, but I can see if I can include one at the end. Now, anyway, so that's what I'm going to say about PTC. Now we're going to take this information. So we're going to take our information we have from our people right here, and we're going to see if we can work with that. Now, basically what we've gotten phenotypes. That's what we're describing here. These are all phenotypes. We're going to see if we can take those phenotypes and try to translate them into genotypes. And so the one thing about this trait is that it definitely has a genetic basis, although the tasting part of it can fuzz the line a little bit, can blur the line a little bit. But the genes do tell you either, you can have this gene or that you don't have this gene. So we'll talk about that in the next section. And so I'm going to switch slides. So in order for us to types and translate them to the genotypes, we're going to need the help from a scientist named Reginald Punnett. So if you've ever heard of the Punnett Square, that's who it's named after. And so he was a geneticist and a mathematician, and he came up with a way of assessing genotypes and seeing what the probabilities would be when they're passed on to the young. And so we're going to be working with a Punnett Square. So now, first of all, though, I just want to point out right here on this slide, I've just kind of taken our little taste test group right there, put their results there so we don't forget them. And so now here is a little chart to describe how this all works. So you have your phenotype. That's basically what everyone was experiencing. So some people are going to be super tasters, some people are going to be partial tasters, and then some people are going to be non-tasters. And so now remember, you get these genes from mom and dad. So you're going to get one gene from mom, and you're going to get one gene from dad. And so I consider if you're a super taster and that PTC tastes super awful, that there's a possibility that you could be a genotype where both parents are tasters. So that's why we have these capital T's right here. So therefore, capital T would mean that that is a tasting gene. So you taste it. If you taste it at all, you're going to get a capital T somewhere. If you don't taste it at all, so where you don't receive a gene that allows you to taste it, that's going to be a lowercase t. So that would be non-taster, not necessarily a non-taster, but just the gene, would be the non-tasting gene. And so when you go to the genotypes, remember, you're inheriting this from mom and dad. So a super taster would inherit 2 tasting genes. So 2 tasting genes for that one. Now, on the other hand, the partial taster, that's someone who can taste it, but it doesn't make their eyes water. And so if it doesn't make your eyes water, then you are considered a partial taster. So there's a possibility that you have one gene from one parent that makes you a taster, and that the other gene coming from one of your parents does not contain the gene. So it's only one tasting gene as opposed to 2. It doesn't mean that one of your parents can't taste it. It just means that they don't have 2 tasting genes. So they had one non-tasting section that they gave to you. Now, in the case of the non-taster, you have 0 tasting genes. There we go. So you're just missing the section of DNA. And actually, you'll see that I don't talk a lot about the PTC section and what they actually test for. And that's because there is actually another course that we have where they actually test for PTC genetically. And so we might even present it. So I don't want to give away information before it's presented in the future. So keep your eye out for a lab about PTC. So you never know, it'll be a higher level lab. It's fun. But anyway, so there we go. So we have, if you're a super taster, you're going to inherit 2 tasting genes, one from mom, one from dad. If you're a partial taster, you're going to get 1 tasting gene from one of your parents. If you're a non-taster, neither parent gave you a tasting gene. Again, it doesn't mean that your parents are non-tasters. They're just partial tasters or non-tasters. So it depends on their genotype and depends on their phenotype. If they can taste it, well, yes, that might mean that they're partial tasters. All right, so now let's take a look how we can work with this Punnett square. So the way he works it out, he puts the mother's genes, so meaning what she inherited from her mom and dad, on one section, and sort of like a multiplication table. And then for dad, what he inherited from mom and dad on one side. So I'm going to go back to my little taste test. Now me, again, my eyes don't water. And so I'm the mom in this case. And so I'm going to call myself a partial taster. And I'm going to say that I definitely can taste it, but I can't taste it too well. So I have one non-tasting gene from one of my parents and one tasting gene from one of my parents. Now my husband, on the other hand, he said it was pretty strong. For him to grimace, it's quite an intense reaction. And so I'm going to call him a strong taster. And so now the way the Punnett square works, just like a multiplication table, you're going to just kind of bring these letters over and down. And so that means in this first box right here, you're going to have a capital T and a capital T. So a capital T from mom and a capital T from dad. Now in the second, I'm a partial taster. So you're going to have one capital T from dad and one lowercase t from mom. Okay, so far in those two boxes, we have one super taster and one partial taster. Now we go down here and we get a big T from dad, big T from mom, another super taster. And then over here, we get a big T from dad and a little t from mom. Okay, so now those are the probabilities of our offspring. And so, that's out of 100% of all of our offspring, that's the probability. So it means that, we have, if you look, we've got right here, we've got two double T's, big T's, two TT's right there. And so then right here we have big T, little t, big T, little t. And so we have a 50% chance to be a super taster and a 50% chance to be a partial taster. But what's the percent chance that we're going to be a non-taster? Zero. 0%. Now wait, I have a problem with that. And the problem I have with that is named Alex, because yes, he is my son. He wasn't switched at birth. It's true. He is my son. Looks just like me, actually, in the male form. And so he's a non-taster, and he's tried this more than once. That wasn't actually first initial reaction that you saw there. He's tried this because he wanted to know what it tasted like, and because he can't taste it. And so he cannot taste it. So therefore, he doesn't fit in to this story right here. So something's got to be wrong. Somebody's got to be wrong. Now, when we're dealing with phenotypes, this is where things can get fuzzy. This is where I said it could blur the line. Because if somebody thinks they're a strong taster, they might not be. And they might just think it seems like it's strong to them, but it might not be as strong as the strongest taster might be able to taste it. So let me change my results a little bit. So I'm gonna erase some stuff here. I'm gonna erase this T. I'm gonna say maybe my husband Mike wasn't sure about whether or not he was really a strong taster. Maybe he was a partial taster and just it was a little more intense for him. Maybe he just ate something that amplified the taste. We don't know. So I'm going to say that he has a little t there. Now let's see how this changes things. So it really doesn't change anything here. So this is big T, same thing. This one is going to be big T, little t, same thing. And then we go down here we've got big T, little t. So, and then we go in this corner and we've got little t, little t. So now that changes all of this. Now, none of this, well, actually some of it is right, but I'll just rewrite it anyway. Some of these are no longer correct. And so the ability to be a super taster is now there's only a 25% chance of being a super taster out of 100%. So for our children, in this case, 25% chance we could get a super taster. So Emily, she said she's a strong taster. We don't know if she's strong or if she's partial. You know, you have to kind of be her. She did want to run away and like wash her tongue with soap. So there's a chance. And there is a possibility. So she could be this TT right here. So there is a 25% chance that she could be that. Now, to be a partial taster, there's a much better chance. There's a 50% chance to be a partial taster. 50% chance to be a partial taster. And to be a non-taster, there's another 25% chance. So now, finally, Alex gets himself on the Punnett Square. And so now this fits a lot better with my family. So it looks like there's a chance that not only am I a partial taster, but my husband is a partial taster too. In fact, that's the only thing that makes sense. And then, so therefore we have a 25% chance to have a super taster, which we might have one. And we have a 25% to have a non-taster, which we definitely know that we have one. And so it's really interesting. And so that actually concludes my little lesson on human variation. I hope you had fun. I hope that you might one day take that little wheel out and try it out on your friends or your class or something like that as a little school project. It might be fun. Something, at least in these quarantines, you could probably do it online or over the airwaves, basically. Now, I did promise you that I would show you some transparent fish. So here we go. I do have some here. And those aren't my personal transparent fish, but these are genetically engineered. So not only are they genetically engineered to not have pigment, but they're also genetically engineered to make, let's see if I can get a color on here. There we go. To make different types of fluorescent proteins. So they make fluorescent proteins that glow under UV light, and they also are transparent. So those are two genetically linked traits, basically. And so fluorescent or fluorescent colors, not fluorescence, not exactly. So fluorescent colors or fluorescent proteins, actually, that's what it is that they're making. And they're transparent. So the fluorescent proteins don't have to do with like melanogenesis or color or pigment exactly. It's a little different. So it works a little bit different. All right, so I hope you enjoyed this. And if you have any comments, of course, I didn't tell you this earlier, but of course there's a chat where you can chat in, and so there will be a moderator there, and to answer as many questions as we can get to. And also, if you have any questions at the end of this, then feel free to put them in the comments, and because we do check our comments and we will try to answer them the best we can. And so have a great day and join us again next time for DNALC Live. Have fun.